Hello everyone, let us get started for the day. The PPT is visible, right? Everyone can see the PPT. Okay, so welcome to the week 12 of the PMRF-NPTEL session on molecular biology. Today we are going to go through the applications of molecular biology and learn how all the techniques that we have used in cloning as well as expression systems are going to be used in different real life applications and industries in order to make our life easier. So we will go through individually all the steps of how different enzymes are used in different applications in industry, such as the milk industry or the food industry, or even in vaccine preparation. So I will quickly introduce myself. My name is Anuvab Chatterjee. I am a PMRF fellow at IIT Kanpur, and I am taking these PMRF NPTEL courses. And you would know that how we do this is that I will show you a question from the previous year's run of the course and then ask you to try and solve it. And then we will try and solve it together and have a better understanding of the concepts behind the questions as well as how to answer these questions in the quizzes that we face during the final exam for NPTEL. So, does anyone have any questions from this week's videos and course? Okay, it seems no one has any questions. So, we'll just get started for the day. The first question that we have for today is blank hydrolyzes the specific peptide bond to generate para-casein and macropeptides in production of cheese. The options are amylase, lactase, protease, and lipase. You can put whatever you think is the correct option in the chat. So these set of questions are going to be pretty important for in MCQs because these are essentially, you need to know this, you can't figure this out. And there are multiple slides on this in the lecture notes as well. So you will be getting asked questions such as does this enzyme, what is this enzyme used for, or what enzyme you will use for this particular application, and you should know how to answer that. So this is one of those questions. Blank hydrolyzes the specific peptide bond to generate para-casein and macropeptides in production of cheese. And you can put in whatever you think is the correct option in the chat. Okay, Chanda and Srijani both say it is lactase B. So let us look at it. So we have been studying molecular biology on a theoretical level for some time now. It has been 11 consecutive weeks in which we have been learning and how the basic fundamental techniques of molecular biology. And today we are going to learn about the various application of molecular biology, how it is used in various meat industries or vaccine preparation or drug delivery, agriculture, or generating genetically modified organisms. The first thing that we need to know that I want to emphasize on is that we have been talking a lot about plasmids. The plasmids that we have been talking about mostly are cloning plasmids. There are various different types of plasmids. The major one of the major classifications divides plasmid into three types. First is a cloning plasmid, then we have a expression plasmid, and then we have a cloning vector, a expression vector, or a shuttle vector. We are going to discuss the difference between the two first, so we have an idea on how exactly we are expressing these different enzymes for our different purposes. First is the cloning vectors. These we have discussed in depth. So all of these have something known as the ORI site, which is the origin of replication, which allows the plasmids to replicate autonomously. So they will keep on replicating. It doesn't matter if the cell is dividing or not, but these guys can keep on replicating. Then we have an antibiotic resistance gene, which helps in selection. So if you have a particular plasmid that can grow in ampicillin, then you grow the bacteria in ampicillin containing media so that any kind of contamination cannot come into play because you're selecting for antibiotic resistance. So ampicillin resistance. So any bacteria that has this plasmid will only be able to grow in that particular media. Then you can have any other selectable markers such as you want the bacteria to produce GFP, so you see green colored cells. Then you have a promoter enhancer, which is typically a strong enhancer such as a CRAG enhancer, which will keep on producing a lot of your gene. And finally, you have your gene itself. So what are cloning vectors majorly used for? These are used in order to make multiple copies of your inserted gene. So if you just have one plasmid that you have made with one copy of the inserted gene, you put it into your bacteria grows and multiplies, and the plasmid is within the bacteria is replicating at a very fast rate. And then you have multiple copies of your plasmid, so you can isolate the gene from there. But cloning vectors are majorly designed to only produce multiple copies of the gene. So essentially, you will have multiple copies of the plasmid. But what do you do if you want to actually express the plasmid? you want the mRNA or the protein from this gene to be expressed in your bacterial system or whatever other mammalian or yeast expression system you're using. In that case, you use an expression vector. The cloning vector just gives you copies and copies of the gene that had initially cloned. Expression vector is what you use if you want to make the gene product express in the bacteria. So expression vectors essentially have typically some more stuff on top of what the cloning vector has. So it obviously has the ORI site, which helps in replication of this expression vector. Then uh, it but also has so the expression vector generally has uh, a promoter enhancer system that you can regulate. So such as the lac operon that we have studied, only in the presence of lactose or a synthetic analog of lactose such as IPTG will the protein be expressed. Then, most importantly, you should have a ribosome binding site. So, in mammalian or yeast systems, this is absolutely important for when the ribosome will come in and sit, and not the mammalian or yeast, but rather the bacterial systems. This is the Shine-Dalgarno sequence, where the ribosome will come and sit and then start translating your mRNA. So a cloning vector at most can produce your mRNA, but expression vector needs to be translated as well. So using the operator and the promoter that you have put in, it will first make the mRNA, and since the mRNA will have a ribosome binding site, the ribosome can sit and then translate your particular open reading frame or coding sequence, so you get the protein. Now, similarly, you should have a transcription termination site which is present so that the mRNA gets terminated at a particular point. You do not want the entire thing to be transcribed, and you should also have obviously have a AUG or a stop codon. Not a AUG, which is the start codon, and the AUG, which is the stop codon, present in the coding sequence so that you have translation working in your system. Then you also have a selectable marker. So essentially, the extra stuff that you have in the expression vector is a operon system that you can regulate, a ribosome binding site, which will be transcribed into the mRNA, where the ribosome will come and sit to start the translation. You should have a translation start site, a translation stop site, as well as a transcription termination sequence. So these kind of plasmids are used in order to translate your protein. So that you actually make your protein. These kind of plasmids are only in order to copy the number of DNA you have. So there is a key difference between the two of them. Now, interestingly, we most of the proteins or many of the proteins that we want to express are actually mammalian proteins because obviously you want to put it into humans. So in that case, you should have a system which can express in the mammalian system. Mammalian expression vectors are also present, which will have mammalian transcription termination sites. And instead of a ribosome binding site, it will not have a ribosome binding site because it will have a 5' capping site where the cap is formed so that your ribosome can sit on that in eukaryotic systems. But the question remains is if you have cloned your particular gene in a bacterial cloning vector, how do you get it all the way over to an expression vector, which is present and can only replicate in mammalian cells? So in between both of these, there is another form of vector called a shuttle vector. A shuttle vector is one which can grow both in the bacterial system as well as the mammalian or yeast systems. So what you do is that you take your cloning vector where you have made a lot of copies of your particular gene, you sub-clone or just take the inserted gene and put it into a shuttle vector, and then you purify the shuttle vector and put it into the mammalian expression vector because obviously you're the changing the organism in between. So you want to go from bacteria to yeast or mammalian cells. So you use a shuttle vector. You subclone to the shuttle vector and then subclone to the expression vector. So most of the time when we are expressing proteins, we are using expression vectors. So how do we exactly clone? We have done this a lot of times now. We've gone through this particular similar slides a lot of times in which we are doing genetic engineering. You use different tools such as polymerases, restriction enzymes, ligases, and the vectors that we deal with. You take your particular gene, you get it from the genomic library or the cDNA library or any other library that you have. You isolate the gene, you add restriction sites to both the ends, use a restriction enzyme to cleave the two ends. You take a suitable plasmid, cut that with the same restriction enzymes, ligate both of them so that you have a vector with the gene inserted, transform into bacteria, and then screen to finally get your bacteria that has your plasmid, and then you grow this bacteria to get multiple copies of your plasmid. And the procedures we have also dealt with, we have dealt with how we isolate genomic DNA, we do PCRs, we do restriction, digestion, and ligation, which is this step. We use competent search for transformation, do the transformation, and then screen using various different techniques. So we have passed all these steps, and now you have the particular gene of interest that you are using that you want to express and then make protein out of. So what is it used in? These proteins are used in various different industries. For example, in dairy industries, different enzymes such as rennet, lactase, protease, or catalases are used for different applications. For example, protease is widely present in the biological world, and it specifically hydrolyzes or cleaves the peptide bond between casein and macropeptides, which is used in production of cheese. This results in the bitter flavor of the cheese and also the desired texture of the cheese that we have. So the solid cheese that we have, we make from milk, you have to put in the protease to make it. And different other enzymes are also used in different other applications, and we will see how they work in subsequent slides. But blank hydrolyzes the specific peptide bond to generate casein and macropeptides in production of cheese. The answer is C, protease. Any questions? Okay, there seems to be no questions for this. So we move on. Next question is maltogenic amylase results in starch granules becoming blank during storage. The options are more flexible, more rigid, more crystalline, or more insoluble. You can put in whatever you think is the correct option, A, B, C, D, in this chat. Jyoti says it is A, more flexible. Anyone else? Chanda also says it is A, more flexible. Rupsha and Oropi also say it is A, more flexible. So let us look at it. So multigenic amylase is also a different enzyme that can be expressed, and you just harvest and isolate the protein and can be used in the a particular industry such as this is also used in the dairy industry as well as in flour production of flour. So it is a supplement for flour. It has an anti-stalling effect and it modifies starch while most of the starch starts to gelatinize, and this results in starch granules becoming more flexible during storage. So you have softer starch granules, so soft bread, that is where you get where you use maltogenic amylase, and this helps in storage of starch as well. So maltogenic amylase results in starch granules becoming more flexible during storage. Next, peroxide is a strong oxidizing agent that strengthens the blank and non-disulfide crosslinks in gluten. The options are ionic bonds, hydrogen bonds, disulfide bonds, or peptide bonds. You can put in what you think is the correct option in the chat. Jyoti says it is C. Jyoti says it is C. Disulfide bonds. Anyone else? Okay, we have multiple answers coming in. Jyoti Ruksha Chandra say it is C. Disulfide bonds. Srijuni and Oropi also say it is C. Disulfide bonds. So let's look at it. So, during production of in the bakery industry, during production of bread, we use an enzyme known as glucose oxidase. So, what does it do? It oxidizes glucose and produces gluconic acid and hydrogen peroxide. But the useful for thing that gets made out of it is hydrogen peroxide, as it is a strong oxidizing agent and strengthens the disulfide bonds and non-disulfide cross-links in gluten. So, in gluten, you have these disulfide bonds that are present. Hydrogen peroxide actually helps in oxidizing this bond even more and making this bond stronger. And it helps in good working conditions and helps in proper function of the bakery system. So, peroxide is a strong oxidizing agent that strengthens the disulfide bonds and non-disulfide crosslinks in gluten. Next, in case of the meat industry, protease enzymes break apart proteins, which disrupts or loosens bland and tenderizes the meat. The options are fat cells, blood vessels, bones, or muscle fibers. What is the use of protease in the meat industry? Okay, Chanda, Oropi, Jyoti, Shruti, and Kasha also all say that it is deer, muscle fiber. So let's look at it. So you probably would have heard about the process known as meat tenderization, in which you want to make the meat softer before cooking it, and the meat is held strongly together by muscle fibers. And you can use something like a protease enzyme such as a protease to cleave the bond that holds the amino acid together. So as a result, the enzyme breaks apart proteins, which disrupts and loosens the muscle fibers and tenderizes it, and you have a softer meat to cook. So protease enzymes break apart proteins, which disrupts or loosens muscle fibers and tenderizes the meat. Next, insulin is a dimer of an A chain and a B chain linked by blanks. The options are hydrogen bonds, ionic bonds, disulfide bonds, or peptide bonds. Okay, you have a number of answers coming in. Srijani Chandra Jyoti and Oropi also it is C, disulfide bonds, and Devadita says it is A, hydrogen bonds. So let us look at it. So we have already gone through a bit of how molecular biology is used in the food industry. Now we are going to look at how exactly it is used in the medical sciences. So medicines are a class of molecules used to correct disturbances in host physiology. They can be chemical in nature and used to inhibit aberrant enzymatic activity from the host or pathogen. In a few cases, host enzymes can be supplied as drug formulation to drive biochemical processes. So essentially, in molecular biology, especially in medicines, molecular biology has an enormous role to play. You use it in production of therapeutically important proteins, gene therapy, vaccines, or molecular antibody production. You'll see one of the most important therapeutically important proteins are insulin. So, a large number of genetic or metabolic diseases can be corrected by supplying proteins or different factors. And in this approach, a gene of an enzyme or a factor is cloned into appropriate plasmid and used to produce a recombinant clone. So, in case of human insulin, so this is known as recombinant insulin. There are different methods of producing insulin. The way the body normally produces insulin is produced as a single peptide which undergoes multiple rounds of folding and post-processing to form the final insulin. But doing that in vitro or doing that outside the body is difficult. So we have used a different strategy. Insulin essentially is made out of the final insulin product. Originally, this is one entire peptide that folds, but the final product is made out of two parts, a pair A chain and a B chain, and both of them are linked together by two disulfide bonds between two cysteine residues. So the way we make it is that we take one of the chains and transform into one bacteria, take another chain, transform into another bacteria, isolate the two peptides, and then combine them together in oxidizing conditions to make these disulfide bonds and finally reconstitute functional recombinant insulin. So insulin is a dimer of a A chain and B chain composed of 51 amino acids and a molecular weight of 5,808. In this process, gene A and B are cloned separately to produce recombinant clones, and peptide chain A and B is overexpressed in E. coli and then isolated and combined together to form functional insulin. Here is another schematic showing the same thing. So, humulin is the commercial name for human insulin, recombinant insulin. So what you do is that you take an expression plasmid with the gene A subunit, insulin gene A subunit. So this only will make the A chain. You link it with beta galactosidase. So as you saw, the entire insulin protein or peptide is only 51 amino acids long. Very small peptides have difficulty in folding properly and forming the correct structure. So and also are very susceptible to degradation because they are essentially you are cutting the original gene in half. So you link it with beta galactosidase so that it forms a bigger peptide which can is actually protected from degradation. You do the same thing with insulin gene B subunit, and after both the peptides are isolated, you chemically cleave beta galactosidase off and then you put them together in oxidizing condition to form the disulfide bonds to have active insulin. When it was actually commercialized in 1980s, was a big breakthrough in diabetes prevention and treatment because previously porcine or pig insulin was used to treat diabetes and used to be extremely expensive, and also because the supply was low because we had to actually get it from the porcine pancreas. But nowadays this we grow this directly in bacteria, and that is why insulin is so dirt cheap. So insulin is a dimer of a A chain and a B chain linked with disulfide bonds. Any questions? Okay. In conjugate vaccine preparation, the bacterial coat is tagged with blank to induce production of immune response against the bacterial coat. What are the bacterial code tagged with? Antibiotics, immunogenic protein, antibodies, or lipid molecules. You can put in what you think is the correct option in the chat. Okay, so we have multiple answers coming in. Oropi, Jyoti, Chandan, Devadita all say it is B, immunogenic peptide. So let us look at it. So molecular biology is also extremely well used in vaccine preparation. Actually, it is the only technique we use to make vaccine. So vaccine is given to develop. So what is a vaccine? Vaccine is a benign or a less harmful form of a particular pathogen or a protein derived from a particular pathogen that is used to build up immunity against the real infection if it ever occurs. So vaccine is given to develop immunity against disease in human or vertebrate animals. Vaccines could be dead bacteria, attenuated or bacteria or pathogens which cannot do the infection, or proteins that are derived from these particular pathogens. There are different strategies to enhance immunological response to give long-lasting protection against disease with minimum adverse effects. So, a quick crash course on how exactly adaptive immunity works. So we have two types of immune response. We have a lot of immune responses. The major being T cells and B cells. So once the body encounters a particular pathogen, there are these T helper cells. These T helper cells go all around the body and actually look at different proteins, peptides, and any molecules that are going around. These have the ability to determine if a particular protein molecule or anything else is inherent to the body or a foreign invading pathogen. Once the T helper cell actually identifies like this is a foreign protein, so that means there is a foreign bacteria going around, there is an infection, then it starts producing a lot of different, uh, like it starts a downstream cascade. The first thing starts doing is that it starts dividing to form a lot of these similar T helper cells, which can identify this particular infection. So once the T helper cell detects a viral fragment, it activates and divides, which is known as clonal expansion. So some of them stay as memory T cells, which will continue. So these memory T cells will go and live in our spleen, and they will continuously make sure that the body remembers what exactly we like. It has already fought this particular infection before, and it has all the tools it needs to fight against this infection later. So some of them become memory T cells, others become killer T cells. These can actually go and directly fight the bacteria and kill it. They also produce cytokines. So cytokines are activating proteins that help the immune system to go into overdrive, and it helps the other type of immunological cell, which is the B cell, to start producing antibodies. Antibodies are these proteins which can actually detect these different foreign bacteria, and these will have the particular this will the T cells will have the B cells produce antibodies that can detect the bacteria it has detected. It also helps macrophages, which are these huge immune cells which can go and engulf the bacteria directly or viruses. So these macrophages are activated, it starts consuming all the bacteria and the viruses. And once all the infection is dealt with, they also produce calming cytokines, or which actually slows the immune processes down that the infection is dealt with. This is how the body deals with the infection. T helper cells recognize a foreign pathogen, then it starts a cascade downstream that helps the body deal with the pathogen. But this does not happen immediately. When the body for the first time encounters a pathogen, then it has a primary immune response in which it starts producing the different antibodies, the macrophages get activated, the killer T cells get activated, and that has a by the time they actually get activated, which is around 14 days, the other part, the adaptive, the innate immunity of the body actually is able to deal with the infection by itself. So, in the initial phase, during the primary immune response, these antibodies and the adaptive immune response play a role, but it is not a very big role. So by the time the primary immune response rolls up, most of the infection is already dealt with. And then most of the response, the immune system goes back down, and then only memory T cells remain in the spleen, which is they retain the memory that the body has already fought with this infection. Then, if a secondary infection occurs from the same bacteria, then the body is completely prepared, and you have a huge secondary immune response. The body immediately goes into overdrive, produces a lot of antibodies. The entire immune system is activated and starts fighting everything it can, so that the infection is dealt with. The secondary immune response is much better equipped to deal with the pathogen that causes the infection. So we have figured this out and use this particular strategy in which we use a vaccine, which is this dead or attenuated form of a pathogen, and invoke a primary immune response. And once the body deals with this weakened infection, whenever we actually get the disease, the secondary immune response is strong enough to actually battle this particular infection. For example, you would have heard of different enzymes such as polio vaccine, BCG vaccine, or more famously, the COVID vaccine. So, all of these vaccines help the body to prepare with the primary immune response to be effectively able to deal with the actual infection with the secondary immune response when the actual infection takes place. So this is why vaccines work. They make sure that the body is ready to produce a huge secondary immune response to fight against the actual infection. So there are different types of vaccines. The ones that were previously used involved killed bacteria. So in killed bacterial preparation of vaccines, pathogenic organism is killed by chemical or UV treatment and used as an immunogen. So the body does not care if the bacteria, like the foreign organism, is alive or not. As long as it has different molecules such as cell surface proteins that it can detect as foreign, it will produce the primary response. So even a killed bacteria can be used to produce a primary response. It is mixed with adjuvant to enhance immunological responses and long memory. Attenuated vaccines in which the organism is treated with chemicals to destroy its ability to cause disease. So this particular bacteria is now not able to produce the disease, but it produces the proteins that can be used to detect it. So again, in order to initiate the primary response. As a result, the organism grows and gives stimulation to the immune system for long-term immunological memory. A toxoid, so various bacteria actually do not cause the disease themselves. For example, cholera, the cholera bacterium actually does not cause cholera. There is a particular toxoid that it releases that actually causes cholera, and that is what the body has to fight against in order to prevent uh, the disease to take place. So in that case, you use a toxoid to create the immune response so that the body deals with the toxin first. So in this vaccine preparation, inactivated toxin compounds are used as an immunogen. Subunit vaccine, as the name suggests, instead of a pure protein that can cause the disease, you take a subunit of the protein and use it to produce the vaccine and have the body recognize it. So, in this vaccine preparation, a pure protein or antigen is given as the immunogen. It is the safest form of a vaccine with minimum adverse allergic reaction because the bacteria itself is not in there to grow and propagate in the body. You just use the protein, and one protein, like proteins by themselves, should not be that harmful, and that is why it is pretty safe. And finally, we have conjugate vaccines. In this vaccine preparation, bacterial coat is tagged with immunogenic proteins to induce production of immune response against the bacterial coat. So you take the bacterial coat from a bacteria, you decorate it with the various immunogenic proteins which attract the attention of the immune system. So you immediately start producing antibodies and the immune response against the bacterial coat itself. So these are known as conjugate vaccines, where you take two different things together, immunogenic peptide as well as the bacterial coat. So in conjugate vaccine preparation, the bacterial coat is tagged with a blank to, to induce the production of immune response against the bacterial coat. So the answer is immunogenic protein. Next, APP mutation to induce peptide aggregation is the key to develop blank in mouse models. Parkinson's disease, Huntington's disease, Alzheimer's, or amyotrophic lateral sclerosis. Okay, Chandra, Ruksha, Koel, and Jyoti, all of them have said C, Alzheimer's disease. So let us look at it. So we are now going to move on to a different application of molecular biology in the preparation of transgenic animals. So what are transgenic animals? Transgenic animals are model organisms in which we introduce different genetic changes so that they can better mimic disease conditions or better mimic different conditions that we can study which are present in humans. So these transgenic animals are used to study underlying disease mechanism, how normal human physiology works, biological products. They are used to test vaccine safety as well as toxicity produced by different drugs. Transgenic animals can be produced in two different ways: by overexpression of a particular gene or knockout or even knockdown of a particular gene. Overexpression is done in which expression of a gene is you want to study if this particular gene has any function. So what you do is that you take the gene and remove it from the particular organism you're studying, say mice, and this works very similarly to an example that I'm going to cite. And so imagine you're going into a room which you have not been to before. You see that all the lights in the room are on, and you have a switchboard close to you. If I ask you to figure out which light is operated by which switch, what is the best way to do it? The best way would be you start switching off all the switches on the board, and whichever switch turns off the light was the probably the switch that used to is used to operate the light. So similarly, to find the function of a gene, the best way to find it is to switch off the gene and see what functions are hampered in the animal. And that way you can figure out what the functions of the gene were. So this is where knockout of a particular gene comes into play. We also call this loss of function of a particular gene. You remove the gene and see any adverse effects, and whatever changes you observe, the gene probably did the opposite of it and made sure that the normal animal could function. So removal of a gene expressing a functional gene, and it's useful to understand the role of a gene. The other side of the coin is overexpression, where you want to see if producing more of this gene has a particular effect, and the expression of the gene into suitable plasmids you or suitable organs is done and can be used for organ transplant. The major studies deal with disease models, in particular transgenic animals. So normally, if you want to study diseases, you have the following conventional methods of studying them, which are naturally occurring genetic mutations. Say a particular person has a type of cancer, you look at the genetic mutations that cause the cancers. You chemically induce the disease conditions in mice or some other organisms, such as streptozotocin-induced DM. You do different physical methods, such as you can do ablation of different nerves to study peripheral nerve injuries, or you use radiation to figure out how the organism deals with it. But the limitations of this is there is no precise control because all of these are macroscopic methods. There is a gross change in phenotype of the organism, and the contribution of multiple organs or multiple systems can take place to help recover from the physical change that you have done. But transgenic animals can counter all these limitations because these animals have the disease on a genetic level, which would be what you will see in humans that have genetic diseases. So you can study it much easier. So, so genetically altered animals or GMOs, which are made from overexpression or knockout of a particular gene, resemble the signature pattern of human disease conditions. So the major advantage being there are target specific genes. You do not need a human subject, you just need the organism, which could be a mouse, to study it. It is cheap and quick compared to getting human subjects, and it is very easily reproducible. And we have made a number of disease models to test out drugs as well as find the mechanism of how the disease works, such as we have the AIDS mice, the Alzheimer's mice, the oncogenic mice. We have already made disease models for diabetes as well as zebrafish based fat. So one example that we are going to deal with is Alzheimer's. So Alzheimer's is a disease in humans, which is a neurodegenerative brain disorder, which means that neurons actually, especially the ones that deal with memory, start degrading over time, and at older age, people start forgetting and cannot remember half their life. So that is why Alzheimer's is a pretty dangerous disorder that happens especially in older subjects, and this occurs by formation of amyloid plaques. So what are amyloid plaques? There is accumulation of particular proteins in the neurons, and since neurons are one of the only cells in the body which do not regenerate or either even replicate, so whatever damage they have accumulated from formation of these plaques, the amyloid beta peptide plaques, remain in the body, and that is why the neurons slowly die. So amyloid plaques or amyloid beta peptide fragments of which are fragments of beta amyloid precursor protein or APP. Accumulation of this protein causes Alzheimer's. So APP mutation to induce peptide aggregation is the key to develop the AD mouse model. So we want to replicate this particular accumulation of beta amyloids in mouse so that we are able to better study it, because in humans you can't open a human up and see what the neurons are doing, but in mice it is pretty easy. So TG2576 is a good transgenic mouse model to start with in order to produce genuine human amyloids, and this was the world's first Alzheimer's mouse incorporated with an APP mutation. The pathophysiology is similar to in the transgenic mouse and is comparable to humans. So any drugs that you make against Alzheimer's in the mouse probably works on humans, as well as any uh, discoveries you make in the pathway of the pathophysiology of the disease also should be translated to humans, and that is why you study it in mouse, which is easier to study, cheaper, and you don't have to pass a lot of hoops to do it, and you then translate the findings to humans. And that is why these kind of mouse models are very, very important. in disease studies. So APP mutation to induce peptide aggregation is the key to develop blank in mouse models. The answer is Alzheimer's disease. Okay, blank are either mutated to induce spontaneous cancer formation. The options are tumor suppressor genes, oncogenes, ribosomal RNA, or mitochondrial genes. Okay, so Aurabi Chanda both have said B, oncogenes. Jyoti says it is A, tumor suppressor genes. And Jyoti changes her answer to B. Khadija, Koel, and Ruksha also it is B. Oncogenes are mutated to induce spontaneous cancer formation. So let us look at it. So this is our very own skid mice. These mice have a very the immune system in these mice are majorly like absolutely removed rather, and that is why they look like this, and they can get infections very easily, and they are protected very much to prevent them from infections from us. And they are also used to study cancer because they can easily are very tumorigenic, so these are used for studying cancers. So cancer mouse models to study cancer formation and to screen anti-tumor drugs, these particular mice are used. So oncogenes are mutated to induce spontaneous cancer formation. So what are oncogenes? So these genes are ones that once mutated can easily give rise to cancer. These include genes which are involved in cell proliferation, DNA damage responses, apoptosis, all of these which actually prevent the cells to proliferate uncontrolled or unchecked. And these oncogenes, when mutated, actually give rise to cancers. Or you can also knock out tumor suppressor genes. So if you remove tumor suppressor genes, then you will also have cancers. For example, HER2, which is a gene, HER2 overexpression is associated with cancer formation, and you have a HER2 transgenic mice, which is an FBB background. This will be a very aggressive mouse. So, and then whenever you activate this particular mutation, then the mice will start having tumors. And various different strains are engineered. Nowadays, we have a billion strains, and you also study oncogenic compounds. Then you can take these skid mice, which easily develop tumor, inject them with uh, cancerogenic, carcinogenic substances, and they will form tumors in that location. And then you use different drugs to treat the cancer and show that yes, the tumor has receded or become more aggressive. So this is how you study cancers in mice. And what are the different advantages of using transgenic animals? So gene requiring certain cellular mechanisms to help with the production of protein. Animals used in transgenic purposes generally carry the mechanism to need to produce complex proteins, and these mechanisms are absent in cell culture. So different parts of the body obviously produce proteins specific to that part of the body, and maybe these proteins can show an effect on the overall organism, but not on individual cells. And that is why you should have transgenic animals to study all of these. Expression through cell culture or bacterial culture requires constant monitoring and sampling. Also, there are not a lot of processes do not happen in cell culture. So, cell culture is when you grow individual cells or layer of cells. But if you want to, for example, study brain development or disorders related to brain development, you obviously cannot study it in cell culture because those are just cells. They will not form the neural tube or form neurons or anything resembling the in vivo structure. So for that, you need organisms or transgenic animals to decipher all the mechanisms behind this particular disorders. Isolation and purification of expressed protein in conventional method is more difficult than purifying proteins for animals' milk or body fluid. It is cost effective to as the product is efficiently passed through milk with an average yield of 53% and a 99% purity. So I do not know if you have heard recently, not recently, sometime back, scientists were able to produce spider silk in goats. So goat milk specifically. So they modified a goat so that whenever the milk comes out, it also produces spider silk. And you know the spider silk is much more has a much higher tensile strength compared to steel, and that is why it generally can be used as a material for the production of high tensile compounds. And thus, people are all looking into this. It has been estimated that transgenic animals can produce 100 to 200 million dollar worth of pharmaceuticals in its lifetime. So you can, like for example, you can produce antibodies and a lot of other things as well. Disadvantages of transgenic animals: the transgenic animal project is extremely expensive in a vacuum, but not as compared to other forms of production of these proteins. These are not that expensive. Generation of transgenic animals are expensive. Yes, production of the first batch is very expensive. That is why no one wants to make their own, and they always try to get it from other sources because of long gestation period, little size, high maintenance, and cost of recipient animals. There can be high mortality rate and other deleterious effects on animals used by researchers to create transgenic breeds. It has been observed that transgenic pigs, having enhanced growth rate and efficient feed conversion, exhibit reduced reproductive performance and may suffer from arthritis and dermatitis. Large number of recipients is required for embryonic transfer because of low transgenesis rate. Transgenic food has been produced and offers better productivity in terms of yield and quality. However, there are some apprehension about safety of transgenic foods, which does not make sense. So till now, people sometimes uh, fight against GMO or genetically modified organisms and genetically modified uh, food stuff, such as especially fruits and barley and wheat. These kind of things people try to avoid. But unfortunately, most of the rice that we have and we consume nowadays is absolutely transgenic. We have completely outgrown the normal, natural, wild type rice to the point that no one grows wild type rice anymore because it does not exist in nature. So we are slowly converting all our natural resources with transgenic varieties because these are safer and also grow at a much faster rate. But time will tell how good they are. So blank are either mutated or to induce spontaneous cancer formation. The answer is oncogenes. You can also do the same thing with tumor suppressor genes. If you mutate tumor suppressor genes, the chances of forming a cancer increases manyfold, but mutating oncogenes is the sure short way of getting cancers. Next, blank acts as a glucose sensor in pancreatic beta cells and plays central role in models of insulin secretion. Hexokinase, phosphofructokinase, pyruvate kinase, or glucokinase. Okay, it seems no one has tried to answer this. So let us look into it. So we also have a more transgenic model for diabetes, and in order to produce diabetes in mice, we cause specific mutation in different genes, such as the gene for glucokinase, which is used to detect the amount of insulin present in blood, or a beta receptor knockout or UCP1 knockout, and these can form either acute or chronic models for diabetes in mice, and then you can treat the mice with different drugs that can prevent diabetes, such as GLP-1 inhibitors, and figure out if your drug is working or not. So, blank acts as a glucose sensor in pancreatic beta cells and plays a central role in models of insulin secretion, and the answer is glucokinase. Next. Blank are used to manage bleeding disorders by replacing missing or defective proteins. The options are coagulation factors, anticoagulants, vaccines, and enzymes. Which would you use to study bleeding disorders or manage bleeding disorders? Okay, we have multiple answers coming in. Srijani Chanda, Devarita, Koel, Jyoti, Rupsha, and Khadija also it is A coagulation factor. So let us look at it. So molecular biology is also used in production of therapeutically important proteins. So if particular disease occurs because there is absence of a particular protein in the body, or the protein does not get formed because of some particular reason, then these enzymes can be produced outside the body and put into the person in order to help with it. For example, insulin is a prime example in which, for different reasons, insulin either is not produced in the body, which is type 1 insulin, or the insulin produced by the body is not enough because of various reasons, and that is type 2 insulin. So you produce insulin outside and inject yourself so that your body can stay in homeostasis. Similar things, other diseases which have a similar norm is hemophilia, which is a blood clotting disorder, and you put in coagulation factors 8 and 9 into the body to make sure blood clotting is not affected. Thrombosis, which the clot formation does not occur properly, you put in tissue plasmogen activator. GI tract infection, which occurs many times, you can use lactoferrin, which is a milk-based iron-binding protein, which simulates mm, which simulates the immunological properties of breast milk. Thrombosis, again, you can use human protein C. Emphysema, you can use antitrypsin. Wound healing, fibrinogen. HIV infection, Pro 542. Thrombosis, again, antithrombin 3. Tissue repair, collagen 1. Blood volume, serum albumin. So, depending which protein causes the particular disease, you can just put it into the body and then that should fix it. That is very bare basics of it, but that is essentially what you want to do. For example, recombinant chymosin is used for manufacturing cheese. A non-pathogenic E. coli strain K12 is used for large scale production. The recombinant enzyme is safe to use, costless, and available in abundance. The human growth hormone, it is produced by pituitary gland, and hormone is required for support of growth and development in humans. It is used to deal with dwarfism and other growth-related defects. Recombinant HGH is again cheap and safe. And finally, recombinant blood clotting factor 8. In a normal individual, blood loss from a damaged vessel is prevented by formation of clot, but in case of hemophilia, clot formation is prevented because the various proteins in the clotting cascade are ineffective or mutated. And if the factor 8 is one of those, which is factor 8 deficiency causes hemophilia, which is one of these diseases, then you can produce and supply recombinant factor 8, which improves the disease condition. So blank are used to manage bleeding disorders by replacing missing or defective proteins as well as coagulation factors. Blank is added to infant formula to mimic the immunological benefits of breast milk. The options are casein, whey protein, albumin, or lactoferrin. You can put in what you think is the correct option in the chat. Okay, we have a number of answers coming in. Chanda Devadita Oropishi joined Jyoti also it is D, lactoferrin. So lactoferrin, as we just discussed, it is given to small babies in case they have neurological defects, and it actually helps ameliorate that by mimicking the immunological benefits of breast milk. So it is a protein which has antimicrobial, anti-inflammatory, or anti-carcinogenic properties and helps the body absorb iron, and it functions as a immunological. It has immunological benefits such as it blocks or attaches to viruses and bacteria and disrupts the cell wall, as well as binds to iron, which the bacteria needs to grow, and thus prevents GI tract infections in small babies. So lactoferrin is added to infant formula to mimic the immunological benefits of breast milk. Blank heart valves have been successfully transplanted into human hearts to replace damaged valves. The options are canine, porcine, ovine, or equine. Okay, so Jyoti Rupshachandra European quail, also B, it is porcine. So transgenic animals are also used for organ transplantation. So transgenic animals that are genetically modified to have organs that can be transplanted to humans. In this case, particular, yeah, so there are these particular proteins that a body checks to decipher if this organ or part of the organ or tissue is foreign or not, and these are called the MH class 2, MHP class 2 molecules. And transgenic animals can be designed so that they produce all the necessary proteins or peptides that the body recognizes as not foreign. So these animals are grown so that they do not express any key foreign antigens and can be a promising alternative to human donors. Pig is the only animal ever used for this because the organs in pigs are pretty much the same size as humans and are very similar to humans as well, so they can work very well. Porcine heart valves are successfully transported into human hearts. Livers, lungs, and kidneys are being tested. Recently, I think kidneys were also tried, but yet these studies are still going on. It physiologically closely matches humans and is much less expensive than monkeys and other primates. So, porcine heart valves have been successfully transplanted into human hearts to replace damaged valves. The Doogie mouse is a genetically modified to overexpress blank transgene in cortex, hippocampus, thalamus, brainstem, and cerebellum. The Doogie mouse has overexpression of which of the following genes? We have different answers coming in. Jyoti, Srijani, Rupta, Devadita, Koel, and Chanda. Also, it is C in our 2B. So, what is the Doogie mice? So, these mice are called also called the smart mouse, which have show higher memory capacity and problem-solving capacity. And these particular mice were generated by overexpression of a particular gene called NR2B in the cortex, hippocampus, thalamus, brainstem, and cerebellum. So NR2B gene actually produces a neuroreceptor in the brain that helps in long-term potentiation. So essentially, it is a calcium channel which allows the neuron to be stimulated for longer, and long-term stimulation of the neuron is actually linked to better memory. And that is why NR2B overexpressing mice actually were able to show better results in memory related tasks, and these are called as smart mice. So Doogie mouse is genetically modified to overexpress NR2B transgene in the cortex, hippocampus, thalamus, brainstem, and cerebellum. Next, bovine leukemia virus BLV causes blank bovine leukosis in cattle. Epidemic, endemic, enzootic, or acute. Chanda Ruksha or PM Srijane, also Coel as well. Also, it is C enzytic. So BLV is a enzootic infection, which means that it is always generally present in as a background in the cattle, and it is very similar to endemic infections in humans, where it is always present and there is nothing like a sudden outbreak. So, for example, normal common cold is endemic. So one of the ways of dealing with these kind of infections nowadays is using chimeric antibody, antibovine PD-1 rat bovine chimeric antibody, which is part cow and part rat, which are able to stably be present in the cow and actually help in degradation of these particular VLV virus. So VLV causes enzootic bovine leukosis in cattle. Final question for the day. DNA fingerprinting uses blank loci because these loci are so variable that unrelated individuals are unlikely to have the same VNTR. So, what loci does DNA fingerprinting use? Variable number tandem repeat, single nucleotide polymorphism, short tandem repeats, restriction fragment length polymorphism. You can put in what you think is the correct option in the chat. Does anyone have any guesses for this? Okay, Chanda, Srijani, Khadija, Koel, Jyoti, and Ruksha all say A, VNTRs. So we are touching on the final topic, which is applications of PCRs in molecular biology, and there are billion applications which include molecular identification using primers which are specific. You can do molecular archaeology, epidemiology, ecology, DNA fingerprinting, classification of organisms, genotyping, prenatal diagnosis, mutation screening, drug discovery, genetic matching, detection of pathogens, a billion different things. You can also do sequencing, which are used for bioinformatics, genomic cloning, and which was majorly used in the Human Genome Project, genetic engineering, which is site-directed mutagenesis and gene expression studies. So they are also used in DNA fingerprinting. So each individual has a different DNA profile known as DNA fingerprint. You would have heard that humans, by comparatively, have 99.8 or 98.5% DNA, which is extremely similar to each other, and 0.5% is what is different. But you will also always have heard that DNA testing is used to catch criminals in different criminal associated cases. But how is that done? So we have particular regions in the DNA which are very unique to us. There are different. These are called tandem repeat regions, and one type of tandem repeat regions are known as variable number tandem repeats because these loci are variable And unrelated individuals are unlikely to have the same VNTR. A sample of a blood, hair, root, or tissue left at the crime scene can be used to identify a person using PCR by comparing the DNA of the crime scene with that of the suspect or with the DNA database of earlier convicts. Evidences from decades-old crimes can be tested, confirming or defending people's originally convicted. So you can take the crime scene evidence and using PCRs against these VNTR regions, you can compare and see if all the different regions match, then this is the DNA from the same person. And this region is so random that the same person probably has the same DNA and no one else. And only people in a very close relation, so local family essentially, will only have the same VNTRs. So VNTRs, so DNA fingerprinting is this variable number tandem repeat reach loci because these loci are so variable that unrelated individuals are unlikely to have the same VNTR. So that is all I had for you today, and that essentially officially closes off our molecular biology PMRF-NPTEL sessions. I hope you learned a lot from this, and best of luck for your exams. We will be having an extra session on Tuesday where I will be going through essentially the gist of the entire course. So we will be dealing with different questions from all the 12 weeks of the course, and I will encourage you to try and attend this. And again, if you want to see any of the recorded videos, you can go ahead and look them up on my YouTube channel in the molecular biology playlist. Okay, thank you everyone for joining. I will see you on Tuesday. Thank you.